I am Ashwini from first MSc Botany. Here we are going to see about rhynia. The topic comes under Paleobotany section. This is one of the type of fossil pteridophyte. Rhynia. Division Pteridophyta. Class Xylophytopsida. Order Xylophytes. Family Rhinaceae. Genus Rhynia. Occurrence. Rhynia is extinct plant having two species, that is Rhynia gyn-wageni and Rhynia major. It was discovered by Kidston and Lang in 1917 from Rhynia chert bed of Middle Devonian. These plants were discovered in the form of petrification. Then external morphology of Rhynia. The plant of Rhynia were herbaceous. Rhynia major was 20 cm in height and 1.5 to 6 mm in diameter, whereas Rhynia gyn-wageni was only about 18 cm in height and 1 to 3 mm in diameter. The plant body was differentiated into subterranean rhizome with an abruptly turned upright photosynthetic aerial shoots. Roots were absent, but at places rhizome was provided with tubs of unicellular rhizoids. The aerial shoots were cylindrical and leafless with a tapering dichotomously branched system. In Rhynia major, the aerial shoots were smooth, but in the case of Rhynia gyn-wageni, many adventitious branches were present on the aerial shoot as well as rhizome. These branches perhaps help in vegetative propagation. The tip of the aerial branch usually bears a solitary terminal sporangium which was about 12 mm in length and about 4 mm in diameter. This is the external feature of Rhynia. First one of the Rhynia major and the second one is the Rhynia gyn-wageni. Then internal structure of Rhynia. Transverse section of aerial shoot and rhizome. Anatomically, the aerial shoot and rhizome are almost similar. Tiers of aerial shoot can be differentiated into three parts. They are epidermis, cortex and stele. Epidermis. It was outermost surrounding layer. It was one cell thick and covered by thin cuticle. In aerial shoots, it was interrupted at certain places by stomata, but stomata were absent in rhizome. Next one is cortex. Epidermis was followed by cortex. It is differentiated into outer cortex and inner cortex. The outer cortex was only 1 to 4 cells thick, thin-walled and without intercellular spaces. The inner cortex had large intercellular spaces and its cell had chloroplast. The endodermis and pericycle layers were absent. Last one is stele. The center of the aerial shoot or rhizome was occupied by stele. The stele was a protostele. The xylem was made up of annular tracheids and there were no sieve plates in phloem. This is the internal structure of rhynia. Then reproductive structure of rhynia. The sporangia were borne singly on the apex of some aerial branches. Each sporangium being oval or slightly cylindrical structure with a little greater diameter than that of the aerial branch on which it is developed. A longitudinal section of sporangium shows that the outermost layer was one cell thick cuticulated epidermis. It was followed by 3 cells thick middle layers of thin-walled cells. The innermost layer was one cell thick tapetum. The wall was surrounding a spacious sporangial cavity which was without columella and contained large number of spores. The spores were of same size. It means that rhynia was homospores. In many species, the sporangium contained tetrahedral tetrads of spores, which suggests that they were formed by reduction division. This is the reproductive structure of rhynia. Thank you.